My name is Niokhor Dione. I'm currently a Vice President of Microbial Discovery and Development at AnimalBiome. I'm specialized on microbial isolation. So during my PhD, I was part of the team that developed a method called microbial culturomics, which allow the identification and characterization of the microbial composition of the gut microbiota of human. So we extended this method to many fields, including animal science and environmental science as well. So currently with AnimalBiome, I've developed and designed a department, the Microbial Discovery and Development Department, where we have our core work is identifying microbes that are important for cats and dogs and design products around them. I came across anaerobic system when I was a postdoc back in Canada in 2019. So as I said earlier, we developed a method called microbial culturomics that use a lot of culture conditions to identify bacteria from the gut microbiome. But back in France, we were making those bacteria, we were making those culture medium. So anaerobic system helped me around ready to use media. So to do my culturing now, I don't need to make those media. So I can just get them off of the shelf and use them. And this helps us, reduce the time we spend making those media, but also it helps us identify bacteria that we were not able to identify in the past because we didn't have those media available. So those are some of the products that I use. And the way that also anaerobic system help us is through their custom media making system. So I remember back in the time when I was a postdoc at Stanford University, we were designing, microbial culturing in the context of the microbiome initiative therapy. So we needed to target specific bacteria that we were not able to grow. So we designed some culture media based on the literature, based on our knowledge, and we sent those recipes to anaerobic system, and they were able to make those media for us, which was extremely helpful in identifying some of the key bacteria that we needed. So I've been familiar with a lot of anaerobic chamber, for example, which is the koi, Don Whiteley. I use all of them back in France and also here in the United States. So one thing that I can say about the IS500, for example, from anaerobic system, is the ability of this chamber to not allow oxygen to get in. With other anaerobic chamber, you can when you introduce a material in the chamber, usually there is oxygen that get in, but it can be flushed out over time when you run a cycle. But with the IS500, the ability of the chamber to systematically sense a bit of oxygen getting in is extremely important to preserve the integrity of some microbes that are extremely oxygen sensitive. In another hand, one of the things also that I have been using on Biolog is the panel for identifying microbes. So I used in the past those panels to not only identify microbes, but to identify what are the key element that this microbe need to grow. For example, we had a story where we had a lot of some bacteria that were not growing well. So to know what is the best way to grow these bacteria, we put them on the Biolog plates and identify what are the main chemicals, what are the main conditions that these bacteria need. And behind, we design the specific media, including the pH, the right carbon source, and the right amino acid that this microbe need. And we were able to get those bacteria growing better by using the Biolog identification panel. I think it's very important for us to understand, all the microbes that live in us. For example, there's a lot of companies that develop today microbiome-based therapeutics, but those, solutions are extremely important for the future of disease control. But I think one of the key components to keep the microbiome field going forward is the ability for us to grow those bacteria. We need to be able to grow every single bacteria that live in us to be able to understand what they're doing. And the only way to do that is by, keep culturing them. And I think having a company like anaerobic system that make media for us, that make anaerobic chamber for us, I think will be very important for the future. And I think we need to develop more culture media and more design, more new media to be able to identify all the unknown bacteria and know what they can do for the future.